So KAPA Biosystems was founded in 2006 and is a global life sciences reagent company. We focus on enzyme engineering by utilizing a process of directed evolution with the goal of developing high-quality enzymes for PCR, qPCR, and next-generation sequencing workflows. So briefly, directed evolution is essentially natural selection in a test tube. We use a combination of random mutations and an artificial selection pressure to evolve enzymes with improved properties. In this example, we start with a gene that encodes for wild-type TAC. Millions of random variants are created via mutagenesis, and these mutants are cloned into a vector which are transformed into an expression vector such as E. coli. Individual E. coli are then compartmentalized into individual reactors using emulsion technology. Each droplet contains one variant, PCR reagents, and a selection pressure. PCR is then performed, and only improved variants which can survive the selection pressure will amplify. The process is then repeated with higher concentrations of that selection pressure until a handful of the best mutants survive. The great thing about molecular evolution is that we generate mutants with extra benefits outside of just the ones that we are looking for. For example, our enzymes are not only much faster, they are also more robust and can better amplify through GC and AT-rich sequences. We can therefore take advantage of these added benefits to produce high-quality kits. And with that, I'd like to introduce our webinar speaker today, Jennifer Pavlica. Jen holds a bachelor's in chemical engineering from UMass Amherst and a master's degree in chemical and biomolecular engineering from the University of Pennsylvania. Jen joined KAPA Biosystems over a year ago and brought with her 5 years of NGS experience from her previous time at Life Technologies. She is currently our applications manager in the support and applications department and focused on our new product development pipeline. And today she will be talking to specifically our RNA sequencing portfolio, specifically looking at enrichment. And with that, I'll pass it over to Jen.
Thank you, Rachel. Today's webinar will focus on KAPA RNA Seq with RiboErase and how it compares to traditional mRNA-seq methods, as well as other available ribosomal depletion technologies. Before we begin our discussion on KAPA RiboErase, I want to take a step back and discuss some of the more traditional RNA sequencing methods and what the sequence data expectations should be when using them. Shown here are the intergenic, meaning between genes, intronic, meaning non-coding, exonic, meaning protein coding, and ribosomal RNA reads for a total human RNA sample. As you can see, over 90% of the reads map to ribosomal RNA. Because ribosomal RNA is not generally of biological interest for most researchers, this means that over 90% of the sequencing capacity is being wasted by sequencing molecules that are not of interest. This wasted throughput results in higher sequencing costs to cover transcripts that actually are of interest at the requisite depth. One way to improve sequencing throughput is to remove the undesirable ribosomal species prior to library preparation through RNA enrichment. Here we are showing what that same total RNA-seq data would look like if we bioinformatically removed all of the reads mapping to ribosomal loci. What we see is a very specific ratio between intergenic, intronic, and exonic reads. As in silico depletion does not solve the issue of wasted sequencing throughput due to the presence of ribosomal reads, RNA enrichment is still required to maximize sequence capacity. In mRNA capture, transcripts that are polyadenylated at the 3' end are selected for over other species, including ribosomal RNA, through a poly-A capture process, which leaves mature RNA available for sequencing. In contrast, in ribosomal depletion, the ribosomal material is directly removed from the total RNA sample, leaving other transcripts of interest available for sequencing, including non-coding and precursor transcripts, as well as both mature and immature mRNA, meaning those that are full-length but not polyadenylated. mRNA-seq is traditionally the more widely used method due to its flexibility and fast workflow. However, its use has implications on the resulting sequence data, which should be understood by researchers wishing to use this method. The bar to the far right shows the relative read distribution of a sample enriched using mRNA capture. In comparison to the previously discussed in silico depleted total RNA-seq data, a notable difference is that the mRNA capture process biases the distribution towards exonic reads at the expense of intergenic and intronic ones. This bias towards exonic loci is a direct result of the selection technology used. In mRNA capture, total RNA is combined with paramagnetic beads, which have homopolymer T stretches immobilized on the surface. Through a series of heating and cooling cycles, the polyadenylated 3' end of mature mRNA transcripts hybridizes to the poly-T on the bead surface. Subsequent washing removes the non-polyadenylated species, such as ribosomal RNA, from the sample. The captured polyadenylated transcripts are then eluted from the bead and input into downstream RNA Seq library construction, which includes RNA fragmentation, first and second strand cDNA synthesis, A-tailing, adapter ligation, and library amplification. Now, as with any workflow, there are limitations. In the case of mRNA capture, one of the main limiting factors is input RNA quality. And to the left, we show a simplified version of the capture process, as previously discussed, using a high quality, meaning mostly intact input sample. The selection process works as expected, with most full-length polyadenylated transcripts captured and available for downstream sequencing. To the right, we show that same process, only using degraded inputs. Because of the presence of breakpoints along the lengths of the transcripts in the sample, the selection process only yields truncated products, with much of the 5' information lost. As a result of this workflow limitation, mRNA capture is not suitable for degraded inputs, including FFPE samples. As previously stated, ribosomal depletion is an alternative RNA enrichment method to mRNA capture, and one of its main differentiating features is its resulting sequence data, shown in the bar to the far right. Notably, ribosomal depletion results in an intergenic, intronic, exonic read distribution that is very similar to the in silico ribosomal RNA depleted sample. This gives a more comprehensive view of the transcriptome without the bias towards exonic loci as seen with mRNA capture. This is of particular interest for many researchers as it is the intergenic and intronic regions that contain many non-coding transcripts, which have been shown to affect gene regulation through a variety of DNA-RNA, RNA-RNA, and protein-RNA interactions. Here we are illustrating this point with a close-up view of a locus on chromosome 12, in which a protein-coding gene, prohibidin 2, is located adjacent to a non-coding region outlined in red. It is evident that the non-coding transcript is well covered using a ribosomal depletion workflow, whereas it is completely missed with mRNA capture. Unlike mRNA capture, there is more than one ribosomal depletion technology available. One option is an enzymatic method, as used in the kappa-stranded RNA Seq kit with RiboErase, which uses a combination of DNA probes and RNase H to deplete ribosomal material. An alternative method is a bead capture-mediated depletion, as used in Illumina's Ribo0 Gold workflow, which uses biotinylated probes and streptavidin-coated paramagnetic beads to deplete ribosomal material. In the RNase H-mediated workflow, DNA oligos, shown in purple, designed to target the ribosomal loci, shown in blue, are hybridized to the total RNA input. As a note, transcripts in green indicate non-ribosomal RNA. The hybridized RNA is incubated with RNase H, degrading any RNA participating in an RNA-DNA duplex, which is comprised only of ribosomal material in this case. After a bead cleanup, the enriched RNA is then incubated with DNase to degrade any residual DNA oligos present in the sample. And after a subsequent bead cleanup, the enriched RNA is eluted and input into the downstream library construction process. It should be noted that the downstream library construction is identical for both mRNA capture and ribosomal depletion. The bead capture-mediated workflow also begins with the hybridization of probes, in this case biotinylated, specifically targeting ribosomal loci. The hybridized RNA is then mixed and incubated with streptavidin-coated paramagnetic beads. The biotinylated probes, and along with it the ribosomal material, bind to the beads. The sample is then placed on a magnet, and the supernatant containing all of the non-ribosomal material is transferred to a fresh tube. The enriched RNA is purified and then input into downstream library construction. Now, though both of these methods ribosomally deplete input RNA, it should be noted that they have differing degrees of success. In a study conducted by the Broad Institute, it was shown that an RNase H-mediated ribosomal depletion method resulted in superior performance with degraded samples, including FFPE, in comparison to a bead capture-mediated workflow. Specifically, the use of RNase H showed improved ribosomal depletion efficiency and transcript coverage uniformity. Here we show the ribosomal depletion efficiency data for a variety of 100 nanogram input FFPE samples using KAPA RNA. The table to the right shows the age and relative qualities expressed in terms of both RIN and DV200 for the 4 samples assessed. We'll discuss these quality metrics in more detail later in the webinar, but briefly, low RIN and DV200 values indicate lower quality samples. With respect to depletion efficiency, the RNase H-mediated depletion technology shows effective and reproducible depletion, even with highly degraded inputs. Here, we show a similar assessment using a high-quality human sample and a wide range of input amounts. In comparison to the bead capture-mediated technology of Illumina's Ribo0 Gold, KAPA RNA shows both more efficient and more reproducible ribosomal depletion. This point is further illustrated when viewing the sequence data in the mitochondrial region for libraries constructed using 100 nanograms of high-quality universal human reference, or UHR, a widely used control sample. 2 observations can be made. The first is that KAPA RiboErase, as previously illustrated, more efficiently depletes the ribosomal transcripts outlined in blue in comparison to Ribo0 Gold. The second is that KAPA RiboErase better covers the protein coding transcripts of interest, outlined in red, in comparison to Illumina's Ribo0 Gold, which has extremely low coverage across a number of these transcripts. These results are intuitive when viewed in the context of the two different ribosomal depletion technologies, along with the mitochondrial DNA transcription process. Transcription of the termed heavy mitochondrial strand results in a long polycistronic precursor transcript, which contains contiguous ribosomal RNA, transfer RNA, and messenger RNA loci. And the RNase H method, the DNA probes hybridize to the ribosomal loci of the precursor transcript. RNase H then specifically degrades the ribosomal loci, and DNA digestion degrades any remaining DNA probes, leaving the other regions intact for downstream library construction and sequencing. In the Bead capture method, the probes are again hybridized to the ribosomal loci of the precursor transcript. However, in contrast to the enzymatic method, the hybridized probes bind to the streptavidin-coated paramagnetic beads, and the full-length precursor transcript is removed from the sample, including the mRNA loci. Our theory is that this results in lower coverage of those transcripts of interest. Both RNA enrichment and the total workflow are obviously of importance when talking about metrics of interest, and shown here are those same results for the coverage of a GC-rich transcript, hemoglobin alpha-1. What we're showing outlined in red is the coverage difference between Illumina and our KAPA RiboErase workflow. In the outlined area of red, we're seeing lower coverage with the Illumina workflow. This is also evident when viewing mallet1, a low-abundance, long non-coding transcript, which is covered more evenly using the KAPA-stranded RNA Seq kit with RiboErase. Lastly, to finish out this comparison between the RNase H-mediated and Bead capture-mediated workflows, we'll take a look at some basic sequencing metrics. Shown here are the comparisons between the duplication rates and coverage uniformity CVs of both the KAPA-stranded RNA Seq kit with RiboErase and the Illumina TruSeq stranded total RNA Seq library prep kit. In the cases of 1,000 nanogram and 100 nanogram inputs, the KAPA-stranded RNA Seq kit with RiboErase shows lower duplication rates, translating to increased sequencing throughput. The KAPA workflow also results in lower coverage uniformity CV, meaning that transcripts are covered more evenly across the full length. Now that we've covered the workflow and the resulting data differences between the different RNA enrichment options and technologies, I want to take a moment to discuss some of the important quality control assays that can be performed to ensure that libraries of optimal quality have been constructed prior to sequencing. We'll be covering input RNA checks, RNA input verification, and final library quality assessments. Accurate library quantification prior to pooling and sequencing is of well-known importance for achieving equivalent and consistent equimolar pooling, as well as optimal cluster density. As such, it will not be covered in today's webinar, but it is worth noting, however, that the most accurate method for quantification is a qPCR assay, such as that used in the KAPA library quantification kit, as qPCR only measures appropriately adapted ligated library molecules that are capable of amplification and sequencing, while other methods such as bioanalyzer, nanodrop, and qubit measure all molecules present in the sample. A question we get quite often from customers working with RNA is how to assess their input material quality. Probably the most widely known metric is the RNA integrity number, or RIN. This is an Agilent software assigned value which takes into account more than just the 28S to 18S ratio. It also factors in unexpected peaks found in other regions of the electropherogram, which are indicative of degradation. RIN scores can vary between 1 and 10, 1 being highly degraded and 10 being nearly fully intact. The other very informative metric is called the DV200 value. And simply put, the DV200 of the sample is the percentage of material that is greater than or equal to 200 nucleotides in length. This metric can also be assessed via electropherogram and involves a smear analysis to calculate the fraction of material longer than 200 nucleotides. Shown below is a bioanalyzer RNA 6000 pico assay electropherogram for a high-quality UHR input. It has a relatively high RIN score of 7.8 and a DV200 of 95%, meaning the sample is mostly intact. While both RIN and DV200 are highly informative for high-quality inputs, differences between the two metrics become more apparent when looking at degraded inputs such as FFPE. Shown here are the RIN scores for the two FFPE samples, breast with a RIN of 2.6 and prostate with a RIN of 2.2. Now, using only the RIN scores as a basis of comparison, one could conclude that these samples are of comparable quality. However, the bioanalyzer traces and DV200 scores tell a different story. The breast FFPE material consists of a majority of highly degraded product with only 2% greater than 200 nucleotides in length. The prostate FFPE, on the other hand, contains a smaller but still substantial fraction of material that is highly degraded. However, 54% is greater than 200 nucleotides, making it a higher quality input than the breast sample. The take-home lesson is that the RIN metric is a great tool for determining the amount of degradation in a high-quality or partially degraded sample, but it is less differentiating for highly degraded inputs. In the case of FFPE, DV200 is more indicative of quality differences. During the RNA-seq library construction process, the first potential QC point within the workflow is after RNA enrichment. It is here that the user can verify the success of either mRNA capture or ribosomal depletion, depending on their workflow. This step involves the use of the KAPA CyberFast one-step RT-PCR kit, which has been optimized for rapid one-step single-tube RNA quantification, performing both reverse transcription and qPCR in the same reaction to obtain quantitative information. This is in contrast to traditional two-step protocols in which reverse transcription and qPCR are performed in two subsequent reactions. For the purposes of this assessment, two primer sets are used, one which targets the ribosomal 28S locus and one which targets the mRNA GapDH locus. Please note that these primers were designed for human sample assessment, and if a different primer set is needed based on organism, they should be designed to produce small amplicons which are less than 75 base pairs in length. Shown here are the resulting data for the two primer sets for two samples, one processed using KAPA RiboErase and one processed using KAPA mRNA Seq. Both samples were generated using 100 nanograms of high-quality total RNA. This assay is designed to measure the CT information for the samples post-enrichment in comparison to a control sample. For 100 nanogram input, the control is 100 nanograms of total RNA. As you can see, the total and depleted mRNA CT values are similar, indicating that mRNA has been preserved through the enrichment process as expected. In contrast, there is a significant increase in the ribosomal CT score for the enriched samples in comparison to total RNA, indicating efficient ribosomal RNA removal as desired. Generally, a CT difference of 10 or greater between the control and enriched samples using the 28S primer set is considered successful RNA enrichment. Our last QC point to discuss today is the final library size distribution assessment after post-amplification cleanup. A number of quality indicators can be visualized using a variety of instruments for electrophoretic assessment, including the Agilent 2100 bioanalyzer, as shown here. The first profile shows an expected library distribution, indicating a high-quality library. In contrast, the next profile shows the presence of a large adaptive dimer peak outlined in red. Adaptive dimer is a molecule generated by the ligation of two adapters to one another due to an overly large molar excess of adapter in the ligation reaction. If found to be a problem in your workflow, their formation can usually be mitigated through a reduction in adapter concentration, an increase in RNA input amount, or through the modification of post-ligation cleanups. Generally, post-ligation cleanups are designed to efficiently remove these undesirable small products, but if they're present in large enough excess, these molecules can carry over into the library amplification reaction. Here, they preferentially amplify due to their small size, around 120 base pairs, and can result in higher molar concentrations than that of the desired library product. As these molecules will efficiently cluster during bridge amplification, their presence results in wasted sequencing throughput. If your final library shows a high prevalence of adaptive dimer, it is recommended to perform a second 1x post-amplification cleanup to remove these molecules. This next profile shows an example of banding as outlined in red. The presence of sharply defined spikes in your final library can indicate an elevated concentration of low complexity ribosomal material that will lower your final sequencing throughput. If these are present in your final sample, we recommend verifying that your selected RNA enrichment workflow is appropriate for your sample type and repeating library preparation, paying close attention to the details of your RNA enrichment protocol. It is also recommended that you use the RNA enrichment verification QC step, as this will directly indicate whether or not RNA enrichment was successful. It should also be noted that banding should not be confused with instrument-related spikes created accidentally during electropherogram generation using the bioanalyzer due to either vibrations during the run, particle contamination, or diagglomeration. The last size distribution phenotype I'd like to discuss is the high molecular weight peak caused by overamplification, outlined in red. In library amplification, primers are typically depleted before other components, and when DNA synthesis can no longer take place due to substrate depletion, subsequent rounds of DNA denaturation and annealing result in the separation of complementary DNA strands, followed by imperfect annealing to non-complementary partners. This results in the formation of daisy chains, which are large assemblies of improperly annealed, mostly single-stranded heteroduplex DNA. These species migrate more slowly during an electrophoretic assay and are observed as secondary high molecular weight peaks. However, they are typically appropriately adapted ligated library molecules of the desired length, which are denatured prior to downstream sequencing. Though we are not going to cover library QC number 4, library quantification, in depth, it should be noted that in the case of overamplification, qPCR-based assays, such as that used in the KAPA library quantification kit, are most accurate as they quantify all appropriately adapted molecules including those that are single-stranded. In contrast, quantification with assays using double-stranded DNA binding dyes generally results in an under-quantification of the final library. For specifics regarding any of these QC points, including library quantification, please feel free to contact our technical support team at support@kapabiosystems.com. To briefly summarize, in this webinar, we have evaluated the data and workflow differences between mRNA capture and ribosomal depletion, as well as compared the data and workflow differences between ribodepletion technologies, showing that RNase H method results in higher depletion efficiency and improved coverage uniformity, including that of GC-rich, low abundance, and precursor transcripts. Lastly, we discussed the important QC metrics to determine library quality prior to sequencing, including ways to mitigate and troubleshoot certain phenotypes. Finally, I'd like to acknowledge some key contributors to the generation of the content used in this webinar, which span both the research and development and support and applications teams here at KAPA Biosystems. And for information regarding KAPA's RNA Seq portfolio, including KAPA RiboErase, please visit our website or contact our sales or scientific support teams.
Great, thank you, Jen. We do have a few questions that were typed in throughout the webinar, so we do have a little bit more time, so if you don't mind, we'd like to just ask a few questions. So one question we received was, what species can you use the KAPA RiboErase kit for?
That's a great question, actually, and we didn't mention it in the content here. But currently, the probes designed to target the ribosomal loci were designed for human, mouse, and rat species. So those would be the supported species for that specific product.
Okay, great. We do have a specific question about one of your QC steps related to banding. Can you please explain again what that banding in the adjuvant bioanalyzer trace indicates, and how do you interpret that banding?
Yeah, sure. Let me just get back up to that slide real quick. So shown here, so basically what we would like to see after library construction is a nice smooth peak of your, obviously, your desired length library. Sometimes what we do see is what we call banding, and those are indicated by the spikes that I've kind of outlined in red here. And so ribosomal material, it's a low complexity material, and so when you don't remove it efficiently, things like fragmentation may not be entirely random, and so what that means is that a lot of these molecules are identical to one another, right? So as they amplify, they basically amplify, and it's a single molecule that causes this spiking that we can see in the final library distribution. So like I said, it's not always indicative of our poor rRNA depletion, and sometimes people can interpret spikes that are due to electrophoretic issues as RNA banding. It's a very specific phenotype in terms of kind of a visual assessment, and this is what I would look out for if I'm constructing a library.
But just to reiterate, so banding can occur from missing a specific region? Yeah,
so exactly. Essentially, if there's a portion of the ribosomal content that's not being depleted and it's being amplified in your final library, it'll amplify a single amplicon, so it'll kind of show up as spikes in your final library.
Great. So you just mentioned that KAPA RiboErase is good for human, mouse, and rat. What would be the expected results if you try to use it for other species? Have you looked at other species with this kit?
Yep, that's actually a really great question, and I think the answer depends on how similar your species of interest is to human, mouse, and rat. So obviously, from KAPA's perspective, what we can guarantee results for are human, mouse, and rat species. But if there's overlap or complementarity in the ribosomal sequences between different species and human, mouse, or rat, we would expect varying degrees of success depending on how much those sequences overlap with one another, because obviously the probes are designed to target those specific regions.
And so if you were to use it for a different animal or a different species, is that where you might see the banding, or you might want to look out for the banding?
Exactly, yeah. So especially species that have less overlap between the human ribosomal sequences, we would expect to see more banding, so definitely keep an eye out there. I'd also say to reach out to KAPA support, because we generally can give some good guidance as to what we would expect for differing results using differing species.
Another question is, what are the input requirements for total RNA for the RiboErase kit?
Yep, so total RNA input requirements are 100 nanograms to 1 microgram of total RNA for RiboErase.
And then along similar lines, have you evaluated the kit below 100 nanograms?
We have internally, and we've done it for what we call high-quality inputs. So I wouldn't recommend using degraded inputs below 100 nanograms, but if you have high-quality inputs, you can go lower than 100 nanograms. There are some modifications that need to be made to the workflow, especially with respect to adapter concentrations, potentially cleanups, and then obviously final library amplification cycle numbers.
Okay, and where is that included in our technical guide?
No, yeah, so if you have any interest in going kind of below what the kit is validated for high quality inputs, I would again strongly recommend reaching out to KAPA technical support at kapabiosystems.com.
Okay, great. We have another question specifically about adapters. So are adapters included in the kit, and if not, what do you recommend?
Yep, so adapters are not included in the kit. That is one component that needs to be sourced separately from this library preparation kit. We generally recommend ordering adapters through a reputable oligonucleotide vendor like IDT. There are obviously some specific modifications that need to be made to adapters prior to ordering. And so again, if you reach out to KAPA Biosystems, we can give you some, a nice guideline that tells you how to order these adapters, what specific modifications you should be looking for, and we can point you towards where you can get the sequence information for the adapters of interest.
And then just a final question here. I think our time allows us only one more question. So you mentioned RIN scores. Do you have guidelines for RIN recommendations for things like the RiboErase kit versus mRNA Seq kit?
Yep, so for RiboErase, it's compatible with degraded inputs such as FFPE, so we don't really give qualifications for which RIN kind of valued materials will or won't work. And we do know that your success in library construction does depend heavily on your input RNA quality. So with FFPE we recommend for RiboErase as much as you can put into the workflow, obviously within the specs of the kit, the better the results will be. For mRNA Seq, as I mentioned, one of the limitations there is its use with degraded inputs. So we would never recommend using mRNA Seq with a sample that has, I would say, a RIN below 8. Maybe 7 would be pushing it, but definitely it needs to be a higher quality input.
Okay, great. I think that concludes the amount of time we have for today's webinar. I'd like to thank everybody for their questions, and thank you everyone for attending today.